Welcome to the podcast for day 34. In this podcast, we're going to talk about animal diversity and some of the unique characteristics of animals compared to fungi, plants, and protists. In this podcast for this day, we're going to cover 3 chapters. Now, all you need to know, though, are the points we're talking about in this podcast, as well as what's on the note-taking guide and what we learn in class. So even though it's 3 chapters, it's a very, very small part of these chapters. There will only be this one podcast, and in this podcast, we will cover what we see right here on Objective 1. Okay, let's go ahead and get started. When we think of animals, we can think of animals in many different ways, but there are 3 general characteristics that all animals fall in. The first is that animals are all multicellular eukaryotes. The next is that they lack a cell wall. We know a lot of other organisms, plants, fungi, some protists have a cell wall, and bacteria, but animals do not. Okay, let's talk a little bit about this point right here. Remember that plants were autotrophs. They could make their own carbon structures by bringing in CO2, and they don't rely on eating other organisms to get their carbon. Fungi, remember, were heterotrophs. And animals are also heterotrophs. Now, heterotrophs means that they can consume food such as large carbon structures, and then these carbon structures can be used by the organism to make other structures within the organism. However, fungi heterotrophs and animal heterotrophs are a little different. Remember that fungi, so if this was our fungus here, it would secrete these enzymes to break up large polymers like polysaccharides so that smaller monomers like monosaccharides, like sugars, were released. And then these were taken up into the cell for consumption. Animals are a little different in the way they apply their heterotrophy. So I'm going to draw an animal still as a single circle here. And what they'll do is if you have your food particle here, and we'll draw it the same way, small monomers together forming the polymer. And what the animal cell will do is, so this is our food here, and this is our animal. The animal will consume this food here, and inside the animal cell are the enzymes that once this is inside there will break it up into the smaller monomer structures like glucose. So the big difference is that fungi secrete their enzymes to begin the digestion of the food outside of the cell, and then brings in the smaller structures into the cell. Animals ingest the larger food structures, and inside of the animal, the large polymers are broken down. Now, with all these different animals, and we see them everywhere we go, scientists classify them based upon several different features, and here are just 5 that your book mentions. Body symmetry, and we'll talk a little bit about that. Number of tissue layers there are up to 3 different tissue layers. Type of body cavity, pattern of embryological development, and growth by molting. Let's first begin by body symmetry. We can think of body symmetry as either being radial symmetry that we see on this page, and we can also think of them as being bilateral symmetry that we'll see on the next page. Something that has a radial symmetry means that, as you can see here, you can cut it in many different ways and you end up with the same sections. A key feature to radial symmetry is that there's a clear top. So like this flower pot, you see a clear top, and it has a clear bottom. This sea anemone has a clear top and a clear bottom, but it lacks any other division. For instance, it doesn't have a left side or a right side or a front side or a back side. It is equal on all sides. Now, counter to the radial symmetry, we also have bilateral symmetry. And a lot of animals that we're used to seeing have this kind of symmetry. Humans, for instance, have a bilateral symmetry. This is just an everyday object, a shovel. You can cut it right down the middle, and it has a top, it has a bottom, it has a left, it has a right, it has a front, and it has a back. Just like this lobster here that we've cut down the middle. It creates two identical halves, but we also have some other directions to this lobster. And most animals that we, I shouldn't say most animals, but a lot of animals that we see. We should define these terms here, a dorsal and ventral. Dorsal in this instance here is the top part of the animal. I often think of, the way I always remember this is, for whatever reason, I could always remember the dorsal fin of, say, a shark or another fish. And we always know, if you see like the movie Jaws, right, you have this water here, right? And then you see this nice dorsal fin popping up. So the dorsal is always on the top. The bottom is always the ventral. So underneath this lobster is the ventral. We also have a right and left. We also have two other terms that might be new to you, called anterior and posterior. The anterior always refers to the head part of the animal, and the posterior always refers to the back end of the organism. Another term you should be familiar with is called cephalization, and this is just a general term talking about how this head is developed. Now, this cephalization is really important, particularly when we're talking about bilateral symmetry, because this is a unique feature of animals, that they have some central nervous system. It may not be a very advanced one in some organisms, and it may be a quite advanced one in others, but they do have this, something unique to other organisms that we've talked about to this point, is this some sort of central nervous system, or at least some kind of sensory system located in the head. So cephalization just refers to the development of that head. Another key way that scientists classify organisms is by the number of tissues they have. Not all, well, the three here are ectoderm, mesoderm, or sometimes people call it mesoderm, and finally endoderm. Ectoderm forms the outer covering. It also, during development, will lead to to the nervous system. Mesoderm, this forms the muscles and actually most of the organs. I remember M in mesoderm and muscles here. Endoderm, this lines the digestive tube and also forms the liver and lungs. So again, the mesoderm doesn't form all the organs, but it does form most of them. And the endoderm goes on and forms a few of the other ones, like the liver and the lungs. Don't worry too much about this picture here. We'll come back to this in just a little bit. But here, at the very least, we can point out where we see the ectoderm. Ecto is on the outside. If you're familiar with the term ectoskeleton, that's the skeleton that's on the outside of an organism. So if you're familiar with that term, that might be an easy way to remember ectoderm for you. The mesoderm, this is on the inside here, and it forms the muscles and most of the organs. And then the endoderm lines the digestive system in both these examples. And we're going to come back to these terms in a moment, so we're not going to define them quite yet. Protosome and deuterostome. Now, some animals, as I said, we define animals by the presence of these tissues, one of these three tissues, but some animals don't have any of these tissues. And these animals that don't have any tissues at all, these would be like your sponges and some other organisms. But sponge is a perfect example of an animal that does not have these tissues. Some phyla have two tissue layers, and we'll talk about those as we move forward. And then the most advanced phyla have three tissue layers, such as humans and most of the vertebrates. All right, another way that we can classify organisms is by their body cavity. Now, the body cavity can be defined as the fluid, or sometimes it's an air-filled space located between the digestive tract, like the stomach, and the outer body wall. So here we see the digestive cavity here, the digestive tract, and then the outer cell wall. Now, depending on how this forms, this is how we can tell, we can classify organisms this way. So we either have a coelomate, well, in this case, we're going to start with the acoelomates, and then we'll talk about the pseudocoelomates, and then finally the coelomates. So as the name might imply here, the acoelomates, often the word a means without or lacking, and the coelom is that body cavity. So these organisms, like the planarian here, lacks a body cavity. And so we see our digestive tube here, and then we see the mesoderm here, or what will become the muscle. And you'll see in the next examples here of the coelomates and the pseudocoelomates that you'll see a white area here which forms the body cavity. So these organisms here that are acoelomates, they do not have a body cavity. Let's talk about the pseudocoelomates. Now, in pseudocoelomates, like we see in the roundworm here, you see the body cavity here. I'm sorry, not the body cavity. You see the digestive tract here, and then you see the body cavity here shown in white. This may have fluid in it, or it may just be air filled. And then it also contains this muscle layer, the mesoderm. You can see how it almost appears that this digestive tract is almost floating there. It's not held in place by any other tissues. So this body cavity is lined on one end by tissue that comes from the mesoderm, and then some tissue formed from the endoderm. Now let's look at what a true coelomate is, and we call these coelomates. Now we see coelomates in a lot of different organisms, such as the earthworm. And you can see here that, unlike what we saw in the previous slide, here's our digestive tract here, and it's held in place by this muscle layer, the mesoderm. Okay. In these true coelomates, again, the digestive tract is surrounded by the mesoderm, and the body cavity shown here is also surrounded on both sides by the mesoderm. I'm going to go back to the previous slide to try to point something out. Pseudocoelomates, the body cavity on one end is surrounded by the digestive tract, a derivative of the endoderm, and on the other end of the body cavity, it's the muscle layer, or the part formed from the mesoderm. Whereas here, the body cavity is surrounded by only mesoderm. And the result of this is that this digestive tube, and as well as other organs, are held in place by these tissues. All right, now let's talk a little bit about reproduction and development in animals. A lot of these terms, some of these terms anyhow, won't be new, but some of them will likely be new as well. So animals reproduce by sexual reproduction. There's a sperm and an egg, and when they fertilize, they form the zygote. We've seen that term before, but this is that one-celled embryo that after it's formed through the process of mitosis will form the multicellular organism. Now, early on, and we'll see this on the next slide, early on in the cell cycle, this zygote undergoes rapid cell division, and we call this cleavage. And an interesting thing about this rapid division that we call cleavage, that even though it's dividing many times, there's very little growth. So the zygote and the next several steps of development, the overall size doesn't change very much, even though the number of cells has changed quite a bit. And we'll see that on the next slide. As it continues to divide, part of this large mass of cells will begin to hollow out, and we call that the blastula. And again, we'll see this on the next slide. And then finally, once this is formed, we see what's known as gastrulation, and this essentially means the gut is being formed. Okay. It's forming the gastrula, which will eventually then become the gut. And at this point, we will get different tissue layers. So here we have our zygote, undergoes massive cell division, and even more so. And even though these are not actual sizes, one thing that is interesting here, all the way through this point here, the cells themselves, when they're dividing, they're not necessarily growing. Those G1 and G2 phases are essentially held in check, and they are very, very short. The size of this blastula, and really even this gastrula stage, is about the same size as the single cell that began the whole process. Very fast division, and it's the reason it's fast is because the G1 and the G2 are reduced in duration. Okay, again, we talked about the hollowing out of this blastula stage, so you have this nice area here where that embryo is eventually going to form, and then you'll see here, these begin to invaginate, like we see here, to form the gastrula during the event called gastrulation. When this forms, we have clear endoderm tissues forming right here, ectoderm on the outside, which will be the outer covering as well as the nervous system, and then the mesoderm has not quite distinguished itself yet. Now, let's talk about this here, this very opening of the gastrula. We call that the blastopore. Okay, speaking of the blastopore, this is another way that animals can be classified. We have two modes here. The first is the protostome, and in this case, the blastopore that we saw on the previous slide becomes the organism's mouth. So over here we have the protostome, and the blastopore that had formed early on now becomes the mouth of that organism. And we see this in some of the lesser vertebrates like the mollusks and the annelids, or should I say lesser animals. And then we also have Deuterostome development, and in this case the same blastopore, but in these animals it becomes the anus, just as we see here. The blastopore becomes the anus, and we see this in a little bit more advanced animals such as the Chinoderms and the Chordates. Another way we can classify animals is by whether or not they shed an external coat. So we see here in this case here we have this crab shown here, and it's just shed this hard external coat here called the cuticle. As it grows, it has to shed this coat. Now animals such as humans, as they grow, they just create more skin cells and they get bigger, yet they don't have an exoskeleton that they shed. I shouldn't call it an exoskeleton, an external coat called a cuticle. A lot of animals like humans don't shed that. Now I want to spend the rest of this podcast talking a little bit about animal diversity. As we look at animal diversity, we can divide all animals up into these 9 phylum, starting with the least derived animals, the sponges, and moving all the way to the most derived, the chordates, of which you and I belong to. With each of these phylum, I'm going to provide a few examples and also talk a little bit about some of their unique features and how they are classified. Some of it's going to be based upon symmetry, some will be based upon the presence or absence of tissues or organ, whether or not they're protostomes or deuterostomes or neither, how they grow. So let's go ahead and get started. So let's begin with the Porifera, the sponges. And we can see here one of the key features of this phylum is that they have no tissues. They don't have the mesoderm, ectoderm, and endoderm. So this phylum for Porifera, again, they make up the sponges. There is no symmetry to them. There's no tissue, no body cavity, no protostome or deuterostome, and there's no molting. So these are the very primitive forms of animals. They don't move, they're sessile. They're filter feeders with intracellular digestion. Now, let's move on to the next one, and we're going to move up in the order of evolutionary appearance. And so we have our Cnidarians here. These make up our jellyfish. So if you've ever seen a jellyfish, or a sea anemone, or a hydra, or corals, these are all Cnidarians. Some of the characteristics of these animals is that they have a radial symmetry, meaning they have a top and a bottom, but as far as ventral, dorsal, left, right, there's no, it's all the same. They have 2 tissue layers. There's no body cavity, so they are acoelomates. They're not protozoans or deuterostomes. We still haven't got to that point yet. And there's no molting. In general, we have 2 variations of the Cnidarians. We have the sessile polyp and the floating medusa. They typically tend to be carnivorous. They have tentacles with poisonous stinging cells called nematocysts. Okay, now let's move to this clade here. They're characterized by having a protozome, yet they still don't have any molting. Okay. A good example of these are the flatworms. And these flatworms belong to the phylum Platyhelminthus. They are characterized by a bilateral symmetry. They have 3 tissue layers, so they have a mesoderm, an ectoderm, and an endoderm. They're acoelomates, so they don't have that body cavity. They're protostomes, and there's still no molting. They're free-living forms. A good example would be the planarians. And then there are some parasitic forms, like the flukes and the tapeworms. All right, let's look at the next phylum, the Annelids. In some of these slides here, I should have removed them. We talk about them belonging to this Lophotrochozoa. Don't worry too much about that. In fact, don't worry at all about that. Okay, just know the names of the phylum that we're talking about. In this case, the Annelids. So let's go ahead and look at some of its characteristics. So in the phylum Annelida, these are segmented worms, like the earthworms that you're familiar with. They're also the leeches and the polychaetes. They also have bilateral symmetry. They have 3 tissue layers. They do have a coelom, so this is the first one we've seen with a coelom. They're protostome development, and there's no molting. They have a closed circulatory system. So that's a new feature we're seeing as well. Okay, so let's look at the phylum mollusks, and let's look at some of its features. These include the clams, oysters, snails, squids, and octopi. Some of the characteristics they have is that they have this bilateral symmetry. They have 3 tissue layers. They have a coelom and their protostome, and still no molting. They have a soft body protected by a hard shell, and the body has 3 main parts. They have a muscular foot, a visceral mass, and then finally a mantle. Now let's move into our first group here, the first clade here, that is a protostome but has molting. So this is the first time we've seen this. And a good example of these are going to be the roundworms, and it's the group called the Ecdyzoa. So I only tell you this name just in case it comes up in a future class, but for the purpose of this class, I'm not going to ask you any questions with the answer Ecdyzoa. We're just going to focus on the phylum. So let's look at the features of the roundworms. There's lots of phylum in this group here. One of the main ones here are the nematodes. These include your roundworms, your hookworms, and your pinworms. These are the hookworm and the pinworms, these are common parasitic worms. Some of the characteristics are bilateral symmetry, 3 tissue layers, mesoderm, endoderm, ectoderm. These are a pseudocoelom, protostome development, and they're the first ones that we've seen that have a molting. Some are free living and some are parasitic. All right, let's look at the next phylum, the arthropods. The arthropods are a very diverse group, probably the most diverse group, and they make up most of the animals we see on Earth. Some of the features of the arthropods is that they have a bilateral symmetry, 3 tissue layers, they have a coelom, a protostome, and they do molt. They make up, as I said before, they're one of the most diverse and numerous animal groups we have. They make up 75% of all animal species. They're a very, very important group to be familiar with. They're segmented animals with specialized segments and jointed appendages. The first time we've seen these jointed appendages within animals. Body is completely covered by an exoskeleton. Now we're going to enter into the last class here of the Deuterostomes. This is the first time we've seen Deuterostomes, and this is the first that we're now entering into chapter 34. Chapter 34 focuses on how vertebrates evolved. And this first group are going to be the echinoderms. These, some examples of the echinoderms are sea stars and sea urchins and sea cucumbers. Some characteristics that are important is that they're bilateral symmetry as a larvae. They have 3 tissue layers. They have a coelom, so they're coelomates. This is the first that we've seen an organism forming the openings through a deuterostome, and we've lost the molting. They're no longer molting. And they are marine. They have a water vascular system for exchange of gas and for movement. And this final group here, the chordates. We'll spend a little bit more time talking about the chordates. They're also part of the Deuterostomia. One of the key features here, or some of the key features of the phylum Chordata, is that they have this notochord. They have a dorsal hollow nerve cord. We see the anus here and a mouth here. They have pharyngeal slits or clefts here. And they have a muscular post-anal tail. So these are all key new features in Chordatas. Some of the examples of the Chordata phylum include the lampreys, the cartilaginous fish, the fish that contain a bony skeleton, and then amphibians, reptiles, and birds, and mammals. The amphibians, reptiles, and birds, and mammals are examples of terrestrial chordates, and the rest are examples of aquatic chordates. I want to spend just a few minutes talking about this transition of going from an aquatic environment to a terrestrial environment, much in the same way that plants did this. Animals had to overcome some challenges as well. The first problem is that of respiration. Aquatic animals used gills to acquire the oxygen from the water, and then as the animals moved to land, they needed to be able to breathe air. So the adaptation that had to form were these lungs, and this helped the animals as they moved onto land to be able to use the air in the environment. And these came from structures in the fish that were the swim bladder. So fish have swim bladders, and these are what evolved into our lungs. The next problem we had was gravity. Much in the same way when we talked about previously, if you see a jellyfish out of water, it has a really difficult time. It cannot resist the pull of gravity. One of the solutions was to develop limbs and a modified vertebrae. These limbs, as we see here, they evolved from the underside lobed finned fishes, the fins located on the underside of these fish. Another problem came with egg desiccation, much like in the plants. So as the animals moved to land, they required some way to protect the eggs against drying out. Remember, in the water, this was not a problem. The solution were these amniotic eggs. And of course, this changed over time between an external egg to an internal egg. But these eggs, nonetheless, were important in protecting the developing embryo from drying out. Okay, that ends today's podcast. If you have any questions, please let us know. We'll see you in class. Bye.